Hi, you all are welcome to today's topic of discussion, which is the male reproductive structures in flowering plants. We know that the kingdom plant is divided into several categories. We have the algae, bryophytes, pteridophytes, followed by gymnosperms, and the uppermost strata, which is comprising of angiosperms. Angiosperms are nothing but the flowering plants, which have developed a very efficient method of sexual reproduction by producing flower and floral parts. We shall today discuss about a typical male reproductive structure which is found in angiosperms. We shall also see the process by which the male gamete or pollen grains are produced inside the male reproductive structure. Flower is one of the basic fundamental structure of an angiosperm. A typical flower comprises of several different parts which are all brought together on a centralized aperture which is called as receptacle. This receptacle supports all the various floral parts which are found in a flower. Before we jump into a typical flower, the first question which arises is how is a flower produced? A flower is produced from a shoot apical meristem, which gets converted into reproductive meristem, which further develops into floral primordia, from which arises the inflorescence axis, on which the floral buds appear, which later on grow to form a mature flower. A typical flower, which consists of a central stalk, which is called as receptacle, which harbors all the other floral parts within it. The first outermost whorl which is found is called a sepal, which helps in collecting all the other inner whorls and protects it. The second whorl followed is the petals, which comprises of beautifully varied colored petals. The basic function of them is to attract insects, birds, and animals and facilitate pollination in angiosperms. The third whorl inner to the petals is the whorl of the male reproductive structure in angiosperms, which is the androecium, or we call it as stamen. And the innermost whorl is where the female reproductive structure or the carpel is found, also known as the gynoecium. We shall further discuss in particularly focus on male reproduction, which is found in angiosperms, which is nothing but the so-called stamen. A typical stamen, two parts. One is a filament, which helps it to attach the anthers together with the help of a connective tissue and attaches the anther to the thalamus of the petal so that it is secured inside the flower. The anther typically consists of two different lobes. Within these lobes, there is a continuous cylindrical structure which is called as microsporangia, inside which the pollen grains are produced. These two lobes are connected with the help of a tissue which is called as the connective tissue. An anther or a stamen consisting of two lobes is called as dithecus. If we were to take a transverse section of an anther, we can see all the internal parts which are associated or found inside the anther in this diagram. Basically, it consists of two layers. One is the microsporangial wall layer, which is the outermost region, and the innermost sporogenous tissue. The microsporangium wall comprises of four different wall layers. The outermost layer, which is formed of thin cells which are equal in size, is called as epidermis. Inner to the epidermis, we find the endothecium, which is comprising of thicker cells, and they may contain thickenings in them. Inner to the endothecium, we find multiple layers of middle layer cells. All these three layers, that is the epidermis, endothecium, and the middle layers, serve as protective in function and protects the innermost microspore mother cells or the sporogenous tissue from the environment. The fourth layer of the microsporangial wall is called as the tapetum. The basic function of tapetum being providing nourishment to the developing microspore mother cells or the pollen mother cells. There is another function of tapetum is to secrete Ubish granules, which are essential for the formation of sporopollen. Please do remember this sporopollen. Come, we'll use this term again in the coming slides. This sporopollen is used for the formation of the outermost wall of pollen grains. The sporogenous tissue, on the other hand, which is found on the inner side, well protected by the microsporangial wall layers, consists of microspore mother cells or the pollen mother cells, which undergo division in order to produce the male gamete or pollen grains. The process by which the microspores are formed from these microspore mother cells is called as microsporogenesis. The microspore mother cell undergoes meiosis, which is nothing but reduction division, and produces 4 haploid pollen grains. Pollen grains are nothing but the male reproductive propagule or young male gametophyte, which are formed inside the anther. They are basically spherical in shape. When first formed, pollen grain is unicellular. The mature pollen grains, on the other hand, contains two cells, a large vegetative or tube cell and a small generative cell. Pollen grains are covered by two wall layers called as sporoderm. The two layers of sporoderm are the outer exine, which is made up of highly resistant sporopollen. This sporopollen layer helps the pollen grains to overcome unfavorable conditions because sporopollen is resistant to high temperature, acidic conditions, alkaline conditions, and so on. Sporopollen may be absent in some regions, and those regions are called as germ pore. It is through this germ pore that the pollen tube emerges out when the pollen grains germinate on stigma. The outer layer or the exine may be smooth or pitted or reticulate or spiny based on the different types of pollen grains that are found in different flowers. The inner layer is called as the intine, is made up of cellulose and pectin. In the newly differentiated pollen grain, nucleus is central and cytoplasm dense. The protoplasm undergoes mitosis, and due to the peripheral position of nucleus, the protoplasm of pollen grains divide unequally into two cells, a large tube or vegetative cell and a small generative cell. The vegetative cell is the largest cell of the male gametophyte, which has an irregular nucleus and large vacuolated cytoplasm and having enough reserve food material. It forms pollen tube that carries the male gametes for fertilization, while the generative cell is the smaller cell of male gametophyte and consists of compact nucleus and thin layer of cytoplasm. It is the generative cell which divides by mitosis to form two male gametes. In over 60% of flowering plants, the pollen grains are shed in two cell stage. When these pollen grains fall on receptive stigma, they germinate, and it is there that the generative cell divides to form two male gametes. While in nearly 40% other, the generative cell divides prior to the release to form two male gametes, and in such cases, pollen grains are shed in three cell stages, that is, containing one tube or vegetative cell plus two male gametes. I hope this topic of male reproductive structure is clear. We have seen the several processes such as microsporogenesis, formation of pollen grains, and the different types of cells of which the pollen grains are made up of. Thank you.